High-resolution melting analysis, or HRM, is a method used to identify variations in nucleic acid sequences. It is the quantitative analysis of the melt curve of DNA fragment following amplification by PCR. HRM experiment generate DNA melt curve profiles that are both specific and sensitive enough to differentiate nucleic acid species.
So how do we apply HRM in pharmacogenomics? The first one is to identify new gene variants. HRM analysis is an alternative to DHPLC sequencing screening of new gene variants. The second one was methylation analysis. HRM can be used to detect methylation differences between genes. This is known as methylation-specific HRM. Mutation scanning. HRM can be used to scan for mutations in target genes for the identification of variant samples prior to sequencing analysis. With HRM, it can scan large numbers of samples for genetic variation. The fourth one is zygosity testing. The higher resolution element not only allows the determination of homo and heterozygosity, it also resolves information about the type of homo and heterozygosity with different gene variants giving rise to different melt curve shapes.
Let's proceed to the advantages of HRM. The first one, it only requires low reagent consumption, which means it will produce only little waste. We don't need to use HPLC solvent or DGGE gels. We only require small PCR reaction volume, which is 20 microliter for each sample in the analysis. And the next one is, it is very simple and fast workflow, which means we don't need additional instrumentation after the PCR application. And the third one, it is a very fast optimization. Unlike the denaturing high-performance liquid chromatography, thermal optimization is not required. And the last one, it requires low sample consumption, and it is very cheap.
So here's come the limitation. HRM is not effective at the intraspecific level because similarity is very high. Different heterozygotes may produce melting curves so similar to each other that although they are clearly vary from homozygote variants, they are not differentiated from each other.
That's all from us.
Thank you for watching!